Parasitic plants are plants that take some of their nutritional requirements from a host plant. The nutritional needs derived from a host plant include organic matter, which is just carbon nutrients, inorganic matter, and water. Parasitic behavior has developed independently 8 times in flowering angiosperms. Roughly 1% of angiosperms are parasitic. Parasitic plants are defined in 3 manners: whether or not chlorophyll is present, where they attach to their host plant, and how they complete their life cycle. Parasitic plants without chlorophyll will not appear green, as chlorophyll is what gives plants their green hue. Parasitic plants have evolved a specialized structure for extracting resources from their host. The ostomium is the main physiological and morphological connection between the parasite and the host. The ostomium taps into the host plant's vascular tissue to extract resources. The origin of the structure is unknown, but it bears many similarities to roots.
Stem parasites are parasites that attach to the stem of their host plant. The example shown here is of a daughter plant. As you can see, it is wrapped around its host stem. Root parasites are parasites that attach to the root of the host plant. Pictured here is a Hydorna africani. Its haustorium is attached underground. Facultative parasites are parasites that do not rely on their host for completion of their life cycle. In other words, this parasitic plant can exist independently of its host if needed. The plant pictured here is Rhinanthus. Obligate parasites require their host to complete their life cycle. A good example of this is mistletoe. Hemiparasites are parasites that only obtain water and mineral nutrients from their host plant. They have chlorophyll to fix their own carbon. Neustria floribunda exemplifies this. Its bright flowers are shown here, and they have no chlorophyll. Holoparasites have no chlorophyll, so they are never green. They take all their resources to survive from their host. The Thailand cyprea is an example of this. Here are some more examples of holoparasites and hemiparasites. As you can see, hemiparasites and holoparasites are mutually exclusive, but obligative and facultative are not. All holoparasites are obligative, while only some hemiparasites are. Facultative plants are only hemiparasites, which makes sense because hemiparasites can fix their own carbon, and fixing carbon is required to be able to potentially complete their life cycles away from their host. Holoparasites must always have a host for carbon fixation, so holoparasites cannot be facultative. They must have a host to complete their life cycle. Many factors are involved in the strength of influence on the host. These factors vary widely, as their effects can be undetectable or even kill the host. There are 4 main factors that contribute to this: the size of the parasite, the growth rate and metabolic activity of the parasite, the degree of dependency on the host for resources, and the stage of development of the host. Factors are interacting. Shown here is Balanophora papuara, a holoparasite. Since they are completely dependent on their host, it would be assumed that their host would die, but they don't. This means that the parasite's size, activity, and dependency are all contributing factors on the strength of the parasite's influence. Because the parasite is using the host for nutrients, and the host is fighting to survive, there is a competition for nutrients, such as water, carbon, and inorganic nutrients. Here is Rhinanthus minor, a root hemiparasite. These types of parasitic plants must compete with their host or the surrounding plants for not only nutrients, but also light. Luckily for these parasites, as well as obligate hemiparasites, moderate levels of light deficiency shouldn't be too detrimental so long as there is an efficient carbon gain. Host-derived carbon is used to fuel parasitic respiration, which is important because parasitic plant respiration is much higher than non-parasitic angiosperms. This lack of carbon can thus hinder vital host processes, such as vegetative growth or flower production. So long as the resource being taken is plentiful, the damage is not too severe. For instance, if the water supply to plants is plentiful, the water diversion to the parasite may be only localized to that area. When it comes to water, root hemiparasites may be able to obtain it from the soil, but stem parasites can only derive it from the host.
Parasites are a sort of end sink for nutrients. Once things are transported to the parasite, then the host will not be able to retrieve them back. Parasites siphon inorganic nutrients from the xylem in 2 ways: through passive diffusion and selective active transport. In passive diffusion, we would see the same ionic concentration ratio in both the parasite and the host. Active transport causes different ionic concentration ratios between the host and parasite. Ionic elements that are mostly immobile, such as calcium and the phloem, are selected for by the parasite, as acquiring them is much harder than the free-flowing ions in the xylem. Despite parasites acting as strong sinks for inorganic nutrients, nutrient deficiency in hosts is unusual, with only some rare cases in host plant growth being affected by parasite sinks. Tobacco plants affected by the purple-flowered Orobanche ramosa saw 30% reduced growth from the 60% reduced phosphorus intake. The host plant's metabolism can be disrupted by all this nutrient stealing. Parasites can induce changes simply by altering the concentration of vital nutrients. For example, xylem-feeding parasites can cause a water loss that reduces the host plant's stomatal conductance, which is the rate of CO2 passage through a leaf. This can cause a fall in the host plant's photosynthetic rate. Interestingly enough, the leaves with the sharpest drop in conductance were furthest from the parasite's hosteriorium. The disruption of the host metabolism is extremely variable, depending on the characteristics of both the host, the parasite, and the environmental conditions. This wide variance makes the exact effects of a parasite on a host's metabolism hard to determine. Parasitic plants are in every major terrestrial plant community, and some aquatic ones too. They are basically everywhere. They can even be found here at UC Davis. Welcome to the suburban jungle, and we've come out here in search of parasitic plants. While they only represent 1% of angiosperms, they are actually quite easy to find. As you can see right here, this is a stem parasite to this tree. We can tell this many ways, one of which is the leaf morphology is quite different between the parasite and the host. Additionally, you could tell that the leaves come in this sort of arrangement and they have a woody stem, while the parasite does not have a woody stem and its leaves sprout in a different arrangement. I'm just going to pull this guy out. Yep, that's parasite. The more you know.